Welcome back, everyone, and welcome to the final session of the day. And our penultimate presenter is Sara Fredriksson, the CEO of Aqilion, who in 2 seconds will be able to speak and actually be heard. Please go ahead, Sara.
So thank you for the introduction and for the opportunity to present Aqilion to all of you today. I know for sure that every member in the Aqilion team have a secret, or not secret, we share it, a dream, a goal, an ambition, and that is to add some new knowledge to how we can stop this vicious cycle of chronic inflammation. So we are a biotech company, and we are approaching this challenge by a few well-selected early drug development assets. So chronic inflammation, they are all lifelong diseases. There is a plethora of them. It affects the whole body. And they share the common theme that they are lifelong. Sometimes they are severe, so they shorten the lifespan. And all of them have a huge impact on quality of life. Now, Aqilion hasn't chosen to work with a specific disease or an organ, but instead we focus on the biology behind the drivers of inflammation. So this is our pipeline and everything we want to do the next couple of years. And I would like to introduce the 3 of these programs to you, just to illustrate how we have chosen them and why we believe that could be a way to move ahead and continue to build value in Aqilion. So let's start with Regulus. In the Regulus program, we are developing a new drug candidate called AQ280. It's a JAK1 inhibitor, and we plan a phase 2 study in patients with eosinophilic esophagitis, or EoE. And I guess that is still a rather unknown disease. Actually, it was first described in the early 1990s. So what is EoE then? It's like an eczema of the esophagus, one could say. So inflammation driven by an eosinophilic influx. That eosinophils, there are cells, immune cells, and they infiltrate the esophagus, it gets swollen, and you have a hard time even swallow. You get pain in your chest. Sometimes food gets stuck. And you can see that if you look at the emergency clinic all around the world, it's quite common that people come in and you have to remove food and sometimes even open up the esophagus a little bit. So we have chosen to work with this indication due to several reasons, and I will show you in the next slide the main reasons from a scientific point of view. Of course, there is a commercial assessment and we can position the drug, but this is a crowded slide. So if you look at or think you're on an immune cell surface. There is an inflammation, it's probably caused by food allergy. And the body messengers of inflammation starts to flow. They're called cytokines. There are receptors on the cell surface, and they recognize these messengers. And then there is a signal into the cell, and there are these JAKs. They are enzymes, and they catalyze this signal that starts the flow of reaction leading to inflammation, fibrosis, and strictures eventually in the esophagus. Now, if you look at dupilumab, that's a biologics. It was approved 2 years ago in the US and now in Europe. And they have shown good effect in EoE. And they can stop 2 of these cytokines, IL-4 and IL-13. But if we inhibit one of those catalyzers, the JAK1, we could actually stop the message from 6 of those cytokines driving this disease. That made us believe, and that's why we're developing the Regulus program. So far, we have concluded a phase 1 study, and we will have some results to present in the next couple of weeks. It's a classical first-in-man study, and we want to find out, is this drug safe, tolerable? Does it have a good pharmacokinetic profile? And can we see that we actually have JAK inhibition? Then we have to follow some biomarkers. And now we're aiming at the phase 2 in Europe and North America. And hopefully, if everything goes according to plan, we will start it in the end of next year. Now, that was an immunosuppressive approach, suppressed inflammation. YERTUB is a little bit different. There we're focusing on ulcerative colitis. It's also affecting a lot of young people, and it has a huge impact of the daily quality of life, of course. So instead of being immune suppressive, we would like to help the colon of these patients to recover its immunostatus. That is, to see that we have those immune cells that actually dampen the inflammation back again, and not just those driving inflammation. And also to get some healing of the mucosal in the colon. So we have identified a small molecule. It's an oral molecule, and we will deliver it locally in the colon. And the target for this drug candidate is another receptor. It's a transcription factor within the cell. But important to remember is that if you are deficient in this signaling, we know that that is one of the causes of IBD. So if we can get that signal back again, then we could promote mucosal healing and also the correct balance of immune cells. So that's why we choose the target. And it's not just to choose the target. Within Aqilion, we work, of course, with a lot of different preclinical assays and studies and market assessment. And most important of all, we would like to see that our partner are interested in the target we have chosen. Now we have all that in place in YERTUB, so we decided to move on with the drug candidate and start to progress towards clinical phase 1 and A and B in 12 to 18 months. So science, I'm a scientist from the beginning, so I have to show you some graphs, otherwise I would feel that I haven't proven anything to you. So don't put too much effort into this one. This is an animal with a disease that is IBD-like, and we treat that animal for 7 weeks with our drug candidate. And then we could show that the colon, it gains weight when you have IBD and it's getting shorter. So if you can lose some weight, that is to get a thinner colon and a smaller length, you have succeeded. That is what you see in the bar to the left. So with our drug, we were able to reverse that event. We were also able in the one to the right to see that the drug target actually binds where it should. So we have a biomarker we can follow the efficiency of the drug. And perhaps the most important of all, we could decrease the amount of cells just infiltrating the colon, and we could see that those cells that actually promote inflammation and start those vicious cycles were decreased as well. Now, Alnitac, the third program. Alnitac was actually out-licensed earlier this year. We made a deal with Merck, and then we had worked with the program for 3 years. And of course, I can't talk much any longer about the development within the program, but I could illustrate why we choose Alnitac and why I believe that Merck was interested in this program. So scientifically again, this TAC1 is also an enzyme. And think of it not as an enzyme, but as a very central switch, so that if you can switch it off, you could actually lower these inflammatory events in many different aspects. So it would match many different diseases as well. We knew that it was undrugged, that was a potential to become the first drug in that class. And we also had some new scientific data. So when we put that together with an idea of chemistry and the feedback from big pharma, we decided to start to make molecules, build IP, and a data package. And that was what Merck actually then wanted to in-license. So today we have a collaborative development work together with Merck, and Merck owns all the global rights to this program. I have said we at least 100 times during 10 minutes, and I'm so proud to be part of this team. This is the Aqilion core team. They are the inventors, they are the project managers, they are the business development people that together build value in Aqilion. And if you like to know more about the team, the programs, and Aqilion, please don't hesitate to contact me. Thank you.
Thank you so much, Sara. So what would you say sets Aqilion apart from your competitors?
That is always a tricky question. I hope and believe that the choice of target and the quality, you cannot always be the first in class, but you can be best in class. And if you match it really thoroughly with those biology driving inflammation, you have an edge. And the other edge is that perhaps we start really early on, identifying target where we know we can find a partner. You have to be a little bit humble as a small biotech. You can't drive all of them into phase 3. You will need to find a good partner. And I think there is always a sense of urgency for new science to reach the patients, and the quickest way is to work with someone that has the resources. Yeah.
So how do you decide from the research, okay, this is worth taking further?
So it starts with the team. Then we have a couple of KOLs, experts. We do a lot of data. They're looking for different data in patent databases, scientific databases. We go out and ask people. We ask Big Pharma what they do, why they didn't promote a project they stopped, and so forth. And if we could say that there are 4 criteria, there should be an interest from Big Pharma, there should be a really potential biology that we can validate behind each target. We should be able to do the molecule without 15 years of research. I think those are the cornerstone, and target is key. Target is everything.
So you, of course, talked about the deal with Merck, and I don't think it's saying too much to say that many companies now look up to Aqilion for your entrepreneurship. So what are the key qualities for successful entrepreneurship, in your opinion?
There are many. The sense of urgency is one, but I think to understand your customer, everything about your customer, and in biotech, your investors. Because if you don't, you can always understand a need. It's not that hard. Medical need, it's easy. But to understand when someone would buy your asset and why, then you're in a good position to negotiate. And sometimes it's too early, perhaps, and sometimes it's not. But we need to be able to show something that can compete and to meet partner requests and also the medical needs, of course. Yeah.
We've had a question here. So from a BD perspective, how many new doors at potential future partners would you say that this deal has opened? What has it meant for you?
So we have closed approximately 20 CDAs with big pharma companies, and then Merck was the one that succeeded all the way. Of course, now, when we approach them to show them our other program, it's a little bit easier to open that door. It was much harder when we were completely unknown, knocking on their door with a preclinical asset. It's difficult.
How long would you say that process was from first starting to talk to Merck to closing the deal?
So if we back one step, I think we started to try and talk with Big Pharma long before we even have data. But the discussion with Merck, it took approximately a year, I would say, from really getting serious until we have closed the deal.
But now you can see that the road is shorter.
I hope so. And we made a lot of mistakes. We made a few good things, learned from them, applied again. And of course, now it's a little bit easier to open the door, but it's still tough competition.
Of course. And as a final question then, could you just tell us what 2024 has in store for Aqilion?
So we are excited to be able to start a phase 2 study, of course, and we want to progress YERTAB as far as we can towards phase 1. And the other big thing during 2024 is probably that we need to finance the phase 2 program, and that's what we're working with right now.
Good, you saved me asking the finance question. So perfect. Thank you so much, Sara.
Thank you for having me.
And we have reached the final presentation of the day, which is from Diamyd Medical, and chair and founder Anders S.N. Möller. Welcome, Anders.
Tack så mycket. Thank you very much. Well, I'm the chairman and the founder of Diamyd Medical, and I'm so proud and so glad to be here, and thank you so much for listening. I'll take this little thing here. These are some of the guys that work in the company, and really our CEO, Ulf Hannelius, he's much better than at doing presentations than I am, but he's in Toronto talking about diabetes, and so he sent me down here, and I'm very glad, of course, that he did that, and it's fun. And this is our disclaimer. Coming back to the story of Diamyd, I like to start with just explaining a little bit, you probably know it already, what is type 1 diabetes. And today, type 1 diabetes starts, it's a progressing disease. It starts with stage 1, it goes over to stage 2, it goes over to stage 3, which is the insulin-requiring phase of the disease. And stage 1 is when you have 2 antibodies, 2 beta cell insulin-producing beta cells. Insulin is produced by the beta cells in the body. And when you have antibodies to these beta cells, you have an autoimmune disease, type 1 diabetes, when you have at least 2 antibodies. The patient doesn't know that he has type 1 diabetes because he doesn't need insulin and the blood sugar levels are pretty normal. The disease progresses, however, to stage 2 type 1 diabetes, and that is when you still have the 2 antibodies, normally to GAD and insulin, and the blood sugar starts to sort of get abnormal, especially after meals and also during meals. So the patient doesn't still know that he has type 1 diabetes, stage 2. So he goes on, although he has abnormal blood sugar values. Then, as the disease progresses and the beta cells get more and more, skadade when you get less and less beta cells, they can't produce enough insulin, and the patient ends up acutely at the hospital, and he gets or she gets insulin every day from that day on for the rest of his life to sustain life. Otherwise, the patient dies. So what we are doing, we are trying to induce tolerance to the autoimmunity. That is, the antibodies is a sign that there is an autoimmune process ongoing to the beta cell producing, insulin-producing beta cells, and we want to induce tolerance to that. So we found, when my daughter got diabetes in the 90s, we talked to several leading KOLs, and they all believed in a molecule called GAD. And we had this Professor Hans Wicksell, who was a professor in immunology and also chaired Karolinska in Stockholm. And he suggested that we should give subcutaneously a prime and a dose and see if we could induce tolerance to GAD and thereby sort of induce tolerance and get rid of the autoimmunity. And so we did, and we did phase 1, phase 2, phase 3, together with Johnson & Johnson. We did rather good results, but they were not significant, and we didn't understand why. Then we sort of looked at how do you treat allergen, and there were research going on where you could give allergen 3 times intralymphatic instead of subcutaneously. And if you give it intralymphatic, you could reduce the number of injections to maybe only 3, and with very small volumes of active product ingredient. So we did that, and we did a phase 1, and we did a phase 2, and at the same time, we sort of looked back at all the data, all the 1,000 patients that we had, and we found that our drug worked in patients that had a certain gene, DR3 DQ2. And that's amazing. I mean, 40% of the type 1 diabetes patients have this gene, DR3 DQ, more or less 40%, at least in the US and Europe. And then there is another major gene that is DR4, DQ8, and they are more sort of insulin antibody driven, that disease. So what we did then was to file patents for intralymphatic injections and for treating the DR3, DQ2, and we started a phase 3 study, and we are right now recruiting in 8 European countries and the States at 60 sites. And we believe that the 300 will be, we already recruited, we think it will be ready recruited. We're fully recruited next year. So that's fantastic. And also, it's not only us, we have a number of scientific papers showing that we're on the right track. I mean, there are so much data now saying that if you have DR3, DQ2, that's a haplotype, that's a certain gene that sort of if they see an antigen, the haplotype molecule sort of catches it and presents it to T cells. And with this haplotype and our molecule, you use a tolerance to T cells. So then it's interesting to see that there is a company called Provention Bio that was acquired a year ago by Sanofi, and it was acquired for $2.9 billion. And 1 year ago, they succeeded in preventing type 1 diabetes from becoming stage 3 insulin requiring. So they gave this sealed anti-CD3 antibody to type 1 diabetes that was in stage 2, that were not really already insulin requiring. And they could stop the insulin requiring phase from sort of coming. And so they got approval for that 1 year ago, and they charge $196,000 per treatment. And the treatment is that you sort of stay in a hospital for 14 days, and you get intravenous injection of this anti-CD3 antibody. So it's a pretty tough treatment, and you also sort of really suppress your immune system totally. You get rid of your T cells, which opens up the possibility to get a lot of side effects. And so that's pretty, people think that is could be dangerous. So it's not something that you really want to do. And what they have done recently, they have also shown that they could prevent beta cells survival in those patients who already have insulin-requiring type 1 diabetes. And they do that by giving this anti-CD3 antibody intravenously twice in 2 12-day courses. So if one course, 14 days, cost $194,000, a 2-course treatment, 2 times 12 days, may cost $300. We don't know that yet, but that's possible. So it's very expensive. So if we look, how does that compare to us? When we do a lot of course, talks to big pharma and other people about our drug and our medicine, and they require a lot of data, of course. So we did go out to a good consultant organization in the States and to try and estimate how much value would U.S. Health Society say that it's worth to have our drug for treatment. And they came up with a figure between $65,000 and $100,000. So I put $80,000 here, and in the States and Europe, there is 50,000 new onset patients with this molecule, with this gene, DR3, DQ2, that present with insulin-requiring diabetes every year. So if you multiply the 50,000 with 80,000, you get to an addressable market to $4 billion. just for that. And then you have the stage 1 and 2 diabetes. That is before you have the open insulin requiring sort of type 1 diabetes, which should add more than 4 billion to the market potential. You have an autoimmune disease called LADA, which is type 2 diabetes. Of the many type 2 diabetes patients, 10% are said to be misdiagnosed. They have like antibodies to GAD or insulin. So 40% of them also should respond to this drug that we have. And then what we have done very recently is that if you give additional boosters, we gave boosters at after 3 years and after 4 years. It's a very, very small study, but we could show that you prevent those beta cells that you have at that time. After 3 or 4 years, you can save even those. So what remains there is, of course, to define the right regimen for a booster, for boosters. That could be once every year, or we don't know that for sure. But we know that boosters will help also. And then, of course, last stage is that once we have stopped the autoimmunity, then we have to regenerate the beta cells to cure the patients. And that might be done by nature itself, by stem cells, or by GABA or something else. So that is really the story behind the company. So thank you very much.
Thank you so much, Anders.
Very nice. Thank you.
We have time for a couple of questions. You have chosen to take the candidate through phase 3 yourselves, which is not entirely common in the landscape of life science, of smaller pharmaceutical companies. What was the reasoning here?
Well, it was just that we saw that it worked, and we had the possibility. So, I mean, we did it twice. First, in 2010, we took it to phase 3. And then at the same time, we negotiated with some of the big companies, and J&J, they sort of we ended up in a partnership with them. And then when we didn't reach significance, we got the technology back, and we restarted with these intralymphatic sort of injections. And then we found out that it worked in this genetic subgroup. So. And then we were on our way. We couldn't stop. So
if we look ahead a bit, what value-adding milestones do you hope to achieve in the coming years, say?
Milestone in the coming years?
Yep.
I mean, first we need to recruit. So there will be some recruitment milestones, I guess. We have, I think we haven't communicated how many we already have recruited, so I won't say that. But, we are recruiting now full speed. And then we hope to have some kind of interim analysis. We don't know exactly what type, and then we have,
And you're also conducting a second study called Diamyd B. Can you tell us a little bit about that study?
That was the one that I very shortly touched on. That's a very small study where we could show that if you give boosters on top of the 3 injections that you give, we could save beta cells even 8 years after the first injection.
And as a final question, then, you are conducting a rights issue at the moment.
Yes.
What are your best arguments for people to participate in this rights issue?
Well, I don't know. To make some money.
And to help.
And to help type 1 diabetes. Yes,
absolutely.
Yes.
That's a very noble cause.
Yes.
Thank you so much, Anders, for your
Thank
presentation.
you. Bye-bye. Thank you.
And with that, we've concluded the presentations and the pitches and the panels of the day, and I will leave the word to Jonas.
Thank you, Cecilia. Well, I'm not going to be long here. I would just want to say thank you to all of you for coming today, and to all the companies presenting, and of course to our fabulous partners who you can meet also in the mingle area outside. We are looking forward to a great program tomorrow as well. We will have lots of presentation and quick pitches, and also a few panel discussions on the themes of financing and, well, finding capital for life science companies, basically. Thank you very much, and I hope to see you tomorrow.